You continue our lesson, seminar work 10, methods of isolation of genomic DNA of plants. Aim of the seminar work is study the methods of isolation of genomic DNA of plants. Plan of the lesson, methods of isolation genomic DNA of plants, and watch the video, plant genomic DNA extraction. The first isolation of DNA was done in 1869 by Frederick Mischer. The literature contains very large numbers of DNA extraction protocols. For DNA isolation, you use usually a combination of physical and chemical methods. The lab equipment and the chemicals and the materials required for DNA extraction: centrifuge, balance, thermoblock, a water bath, vortex, mortar and pestle, micropipettes, extraction buffer, stub or DLC buffer, isopropanol, sterile distilled water or molecule water, chloroform, isoamyl alcohol or isoamyl alcohol, and 70% ethanol, Eppendorf tubes, racks, and tips. Choose of plant material. The process of plant DNA isolation started immediately when the plant material has been sampled. The young tissues is rich in DNA and preferred than the old tissues for DNA extraction. And the analysis of fresh tissues is probably preferred in most cases. If you collect the samples, plant samples in the field must be stored at refrigerator at 4 degrees Celsius before the DNA extraction. Labeling of plant material are essential if you work with large number of samples for analysis. Standard methods of plant genomic DNA extraction: grind the samples, plant material into powder with use liquid nitrogen or any buffer, stub buffer or DLC buffer. Transfer the tissue to tube and add 1 milliliter of extraction stub buffer. Then mix gently and the inversion and heat this plant sample into water bath or thermoblock at 55 or 60 degrees Celsius for 20 minutes. Then centrifuge at 50,000 RPM for 5 minutes. After centrifugation, transfer the supernatant to a fresh tube and add 1 volume of chloroform. Centrifuge at 50,000 RPM for 20 seconds and transfer the upper aqueous phase to a fresh tube and add 1 to 10 volume of ice cold ethanol. Then place a freezer at 20 degrees Celsius for 6 minutes. Control questions. Watch the video, plant genomic DNA extraction.